everyone. Good evening. I hope I'm audible. Good evening, everyone. Please confirm. I hope I'm audible or not. Okay. So, as we know, we have started the Mahamarathon series. So, in this today's class, we will going to talking about your microbiology, which will basically cover your basic introduction to microbiology, microscopy and staining techniques, different culture media, bacteria, virus, fungi. We will discuss most important high yield question for your upcoming GPAT exam. So these questions will be from above topic which is shown in your screen. Good evening all. I hope all of you are doing well. Let's start. So the first question, who were the key figure responsible for the beginning of the golden age of microbiology? Your options are Edward Jenner and Joseph Lister, Louis Pasteur and Robert Koch, Alexander Fleming and or Paul Ehrlich, Anthony Leeuwenhoek, and Robert Koch. So answer this question. So, who were the key figure for the responsible of golden age of era? So if we'll talk about the golden era, so golden era of microbiology basically starts with Louis Pasteur and Robert Koch. Okay, it is basically starts with Louis Pasteur and Robert Koch. So you will see the era basically classified into discovery era, transition period, golden age, and 20th century era. So Louis Pasteur, which is considered as the father of modern microbiology. If we'll talk about modern microbiology, Louis Pasteur is the one. If we'll talk about the father of microbiology, that is the Antony van Leeuwenhoek. Okay. So Louis Pasteur is also associated with one of the important sterilization technique, that is pasteurization. I hope all of you are know. Okay. Next question. Which of the following is not a Koch postulate? So Robert Koch have given four postulate, four important points. So you have to find which of the following is not correct. Okay. So please focus on the term not correct. The first option is the microorganism must not be re-isolated from the inoculated disease individual and matched to the original microorganism. The microorganism must be found in disease but not in healthy individuals. The microorganism must be cultured from the diseased individual. The inoculation of healthy individual with the culture microorganism must be recapitulated in the disease. Answer this question. Hi Abhishek. Answer this question. So which of the following is not a correct one? Yes. So Robert Koch had given four important points. But according to this point is incorrect. Okay, so he told four important points. First point, key microorganism must be present in a diseased individual, not in a healthy individual. First point is correct. Second point is the microorganism must be found in a disease but not in the healthy individual. That first point. Second point is microorganism must be cultured from a diseased individual. It can be cultured in a healthy individual. If you inoculate the microorganism, it can recapitulate it into a disease state. Okay, so this is the four important points. The organism suffering from the disease, the microorganism must be isolated in a grown in a pure culture medium. When the microorganism is introduced to a healthy host, the same person can be converted into a diseased person, and the microorganism can be re-isolated from the experimentally infected host. Next question. Which of the following statement is not true? Okay. Which of the following statement is not true about prokaryote. Okay. So first point, nucleus is not bounded by nuclear membrane. Cell membrane contains peptidoglycan. ATS ribosomes are distributed in the cytoplasm, or it is haploid in nature. Yes, answer this question. Answer this question. Which of the following statement is not true about prokaryotes? So if we'll see, so nucleus is not bounded by nuclear membrane. Yes, that's why it is called that in case of prokaryote, true nucleus is absent. Okay, true nucleus is absent. So here the question focus on not true. Okay, so which is the following statement which is not true? Cell membrane contains peptidoglycan. Yes, correct. ATS ribosome is distributed in cytoplasm. No. So in case of prokaryote, 70S ribosome is there, not ATS. Okay. Option C is the correct answer here. So you can see in case of, if you see in case of prokaryote, you have 70S ribosome. You can see eukaryote 80S. You can see, you can follow our this module number 14. So everything is written over there. Okay. Other also DDC Navigator series, page number 14. Module number 14, page number 129. Okay. Next question. The major difference between a prokaryotic and eukaryotic protein synthesis mechanism in which part of the process? So first tell me, the protein synthesis process is called as what? The process of synthesis of protein is called as what? The protein synthesis process is basically called as translation. Okay. So translation. So this translation process in case of prokaryote and in case of eukaryote are different. In case of prokaryote and in case of eukaryote, it is different. So what is the whether it is different in initiation, whether it is different in elongation, whether it is different in termination. So translation process is consisting of three steps: initiation, elongation, and termination. The major difference between, okay, please mark my word. The major difference between the protein synthesis process in comparison to eukaryote and prokaryote is initiation. The initiation step in prokaryote is easier, but in case of initiation of protein synthesis in case of eukaryote is highly complicated. Okay. Next question. Which of the following statement are true? Okay. This question was asked in GPAT 2022. Which of the following statement is true over here? Bacteria are categorized underneath the kingdom Monera. Okay. Protista are unicellular eukaryotic organism. Yeast and molds are under kingdom fungi. Multinucleated higher fungi are under animalia. You have to find out which of the following statements are true. A, B, C, B, C, D, A, B, or C, D. Yes, answer this question. So bacteria is categorized by under Monera. Yes, it is absolutely correct. Protista are unicellular and eukaryotic organism. Yes, correct. Yeast and molds are work under fungi. Yes, it is correct. But multinucleated higher fungi are not comes under animalia. It is comes under fungi category. Okay, eukaryotic fungi category. So option D is incorrect. So A, B, C is correct. Okay. So kingdom Monera. So we have total five kingdom. What are the name of these five kingdom? One is your Monera. Yes. Another one is Protista, Animalia. Next is Plantae and Fungi. Five kingdom. You can see. Monera, Protista, Fungi, Plantae, and Animalia. Okay. Next question. So you can find all this question in our GPAT marathon module. Okay. Next question. You can join the online test. Whatever the question we are discussing, you can attend all these questions by giving the exam. So you can join the courses by joining our this course. Open the GDC classes app. So in the batch section, you have to apply the code MAHA26. MAHA should be in small letter. So then it will be joined at the test time is 9 p.m. So whatever the questions we are discussing with you guys, if you'll not maintain the stipend, so what, Pratik Satyanarayan, if we don't maintain proper percentage impact, the GPAT stipend will get stopped. Yes, actually, in case of NIPER, you need to maintain at least some CGPA in your examination. If you will not maintain, then you will considered as fail and your stipend must be stopped. Okay, so for that, you need to maintain a proper CGPA throughout your MPharm program. Okay, you need to maintain. So you can join this batch. The batch code is MAHA26. So you can join it. And I want every students to join over there because it is the actual time to practice this MCQs. Okay. Next, the phase contrast microscopy in individual, it is a valuable studying living cells which are stained, unstained, treated with a fluorescent dye or treated with a fluorescent antibody. So answer this question. So phase contrast microscope. Yes, phase contrast. The advantages of phase contrast microscopy is we can visualize, okay, we can visualize unstained biological sample. Okay, this is the advantages. So here the correct answer will be option B, unstained sample. So unstained sample can be visualized. For the stained sample, you need bright field microscope. Bright field microscope. For the fluorescent antibody and fluorescent dye, you need which type of microscope? Fluorescence microscope. Fluorescent microscope. Okay, very good. So you can visualize unstained samples, unstained cells. Next question. Next question. In a bright field microscope, how does the specimen appear against in the background? Dark against bright background, bright against dark background, fluorescent against dark background, three-dimensional against flat background. So first we are talking here about bright field microscopy. When you are talking here about bright field microscopy, the fluorescence option is automatically removed. The three-dimensional against flat background option also removed. Now we have to find out this from these two, one option should be correct. So in bright field microscopy, what happens? We basically use a bright background and in front of bright background, a dark object is placed. So we are visualizing a dark against a bright background. Option A is the correct answer. Okay. I hope everyone is getting the points. So it is optical microscope that utilize light rays microscope that to produce dark image against the bright background. Okay, option A is the correct answer. Okay, next question. Basic dye are used in the microbiological staining. Which of the following statement is wrong about the mechanism of staining? So if you remember, we have three type of stain. One is our acidic stain, one is your basic stain, one is your neutral stain. So if we will talk about acidic stain, it stains the basic part, basic stain stains the acidic part, and neutral it can stain the both part. Here we are talking here about basic dye. We are talking here about basic dye. So basic dye, these are the positive ions on the surface of the bacteria form a color complex with dye. Ion exchange between negative charge on the bacteria and positive dye takes place. Ion exchange between positive charge on the bacteria and negative charge on the dye takes place. The neutral charge on the surface of the bacteria forms a color complex. Yes, answer this question. What is the mechanism of action of a basic dye? So the basic dye, basically, these are the neutral charges on the surface of the bacteria that formed a color complex with the basic dye. Option B is the correct answer. Okay. So another things which can be asked from this acidic dye and basic dye concept is its example. If you see the acidic dye example is acidic dye. I am writing here. If you want, you can write it down. Acidic dye are eosine, nigrosin, Indian ink. This is acidic dye. If we'll talk about this basic dye, so your example is methylene blue, then safranin, then crystal violet. Crystal violet. Okay, malachite green also. And if we'll talk about neutral dye, so neutral dye example is your giemsa stain. An important point about giemsa stain is it is used for the detection of malaria parasite. Detection of malaria parasite. Okay. Next question. Which of the followings are basic stain? Just now we have discussed all. I hope this you all can answer this question. Crystal violet, malachite green, methylene blue, sarfarin, and Indian ink. From which you have to find out which are basic stains. Only Naiper colleges or as normal colleges? I think in I actually I whatever the information I shared with you that is related to Naiper, but I don't know about other colleges. Uh, I will let you know. Okay, I have to check. I have to ask some friends and I will tell you. Actually, I don't know about other colleges, so I have to ask. So answer this question number 9. Which of the following is the basic strain? Basic strain: crystal violet, methylene blue, saffron green, Indian ink. Just now we have discussed crystal violet, methylene blue, and saffron. These all are three are basic strain. But this Indian ink, it is comes under acidic strain. Option C is the correct answer. Next question. So here question is 6. See the question. Which of the followings are the basic stains? Okay. So basic stain is your methylene blue, crystal violet, saffronin. Okay. Your Indian ink is comes under your acidic dye. Next, saffronin is used as a reagent to detect gram-negative bacteria, gram-positive bacteria, acid-fast bacteria, या myxia. Why saffronin is used? So saffronin, whether it is used for the gram-negative bacteria, whether it is used for the gram-positive bacteria, whether it is used for the acid-fast bacteria or used for myxia. You have to find the correct answer. This question was asked in GATE 2004. Okay, this question was asked in GATE 2004. Option last question, option was C. Very good, correct answer. See, in gram staining, the use of saffronin for the saffronin for detection of gram negative. Okay. So now next question can be what another question can be formed from this sapronin. What is the use of sapronin in gram staining? It is used as a counter stain. So if you will see, so we have 4 steps. Okay. One is primary dye. First, you need to apply primary dye. Next is moderant. Next is decolorizer and counter stain. Okay. So primary dye, moderant, decolorizer, and counter stain. So option A is correct here. Primary dye used is crystal violet, methylene blue. Moderant used is grams iodine. Decolorizer is acetone or alcohol. And counter stain is sapronin. So sapronin, use of sapronin as a counter stain. This is there in our module number 14, page number 140. You can refer. Okay. So in the gram staining, what is the use of iodine? Just now I have told you. So there is a mnemonic. So what is the mnemonic? The mnemonic is Prime Minister Delhi Chalo. Prime Minister Delhi Chalo. Okay, this is the mnemonic. Prime Minister Delhi Chalo. What you need to remember? So first thing is Prime Minister prefer primary dye. M for moderant. D for decolorizer. C for counter stain. Okay. So primary dye here is crystal violet or methylene blue. Crystal violet, methylene blue. Second, moderant is used here is grams iodine. Grams moderant is used here is iodine. Decolorizer is acetone or alcohol. Counter stain is saffronin. So here the option C is the correct answer. Okay, next question. During gram staining technique, saffronin and neutral red are used as primary stain, moderant, counter stain, or decolorizer. Just now we have discussed. Saffronin and red neutral shade are used as what? Answer the question number 12. The correct answer here is counter stain. So prime saffronin and neutral red are basically used for the counter stain. Next question. Upon gel Nielsen staining, the acid fast bacteria appears in which color? So first thing, gel Nielsen staining is used for the acid fast bacteria. Why it is used? It is used for the acid fast bacteria. So question also asks, why acid-fast bacteria gives positive test for gel nasal staining? This is because presence of mycolic acid. Due to the presence of mycolic acid in their structure, they show positive test for this staining. Okay. So in gel nasal staining, acid-fast bacteria show which color? Blue color, green color, violet color, or red color? Answer this question. Acid fast bacteria will going to give red color, violet to red color, but red color is the accurate one. Okay. So they will acid fast bacteria. The acid fast bright red to intensive purple color will be shown by this acid fast bacteria. Non acid fast bacteria will going to show which color? It will going to show blue color. Okay, blue color for gram negative acid fast bacteria. A bright red to intensive purple color is for positive bacteria. Okay, next question. A specimen obtained from patient's cerebrospinal fluid, okay, cultured in a specialized medium for 5 weeks, showed the presence of bent rod and it tested positive for 𝘿𝙄𝙎𝙎𝙎𝙬 reagent. Identify the organism. Neisseria meningitis, Mycobacterium tuberculari, Bacteroid fragile, Leptospira intrigans. So this question was asked in GATE 2001. So it is telling that the particular specimen which is isolated from the cerebrospinal fluid as cultured in a specific culture media. After the staining, what happens? So it is so positive test for 𝘿-Neilson staining. So if a bacteria is giving a positive staining for 𝘿-Neilson, that means this is a which type of bacteria? It is a acid fast bacteria. Acid fast bacteria. So example of acid fast bacteria is your Mycobacterium tuberculari or Mycobacterium leprae. Okay, so here the option B is the correct answer. Next question. Acid fastness in acid fast staining is due to the abundance of mycolic acid, squalene, ergosterol, yeah, peptidoglycan. Just now we have discussed this question. If you will see, the mycolic acid is the component. So mycolic acid is the component which is present in the cell wall of acid fast bacteria. Okay, so this is having high lipid content. Okay, it is having high lipid content. So due to this property, they are stained by acid fast bacteria. Okay, next question. So you can order our GDC Navigator series, which is consisting of colored book. Okay, 16 colored book. So 16 modules, which is the cost is around 2,500. If packed up to 32, which contains 16 plus 6 modules, and if Navigator series package 3, which contains 16 plus 6 plus 1, total 23 modules. Okay, you can order it from our GDC classes app also. Okay, let's move to the next question. Which of the following statement is correct? You have to find out which statement here is correct. Fusion is used for the nucleic acid staining. Tannic acid is used for the coating of flagella. There are two reagents. You remember first thing. So one is your fusion. So fusion is used for the nucleic acid staining. That is correct. It is used for the nucleic acid staining. Second point here is tannic acid. Okay, colloidal solution of tannic acid. Okay, if you have we have read colloidal solution of tannic acid. Okay, colloidal solution of tannic acid is basically used for which purpose? It is basically used for the coating. So you can see these two are statement are correct. These both the statement are correct. Both 1 and 2 are correct option. Okay. You can see flagella staining, colloidal silver colloidal suspense of tannic acid and fusion. So these two fusion is staining for basically for the chromosomal staining of DNA, and this fusion silver colloidal sulfur tannic acid it used for the flagellar staining. Okay, flagellar staining. Next question. Giemsa stain is used for the staining of GIMSA stain is used for the staining of what? Neisseria, malaria parasite, mycobacteria, या alin. So this is very, very important question. Just now we have discussed. So first thing is, GIMSA is a which type of stain? Tell me. Answer in the comment section. GIMSA is a which type of stain? GIMSA is nothing but it is basically called as a, yes, neutral stain. Very good. It is a neutral stain. So, or neutral dye, neutral stain or neutral dye. So, this neutral stain or neutral dye, this is used for which purposes? It is basically used for the detection of malaria parasite. Okay. Why it is used? It is used for the detection of malaria parasite. Option B is the correct answer. Next question. Production of acetyl methyl carbinol. Production of acetyl methyl carbinol can be detected by which of the following tests? Okay, so production of acetyl methyl carbinol can be detected by which of the following tests? 𝙂𝙖-Pascal test, indole test, citrate utilization test, methyl rate test. So first thing is, these four are comes under which test? These four are comes under IMBIC test. So why this IMBIC test is used? It is used for the coliform forming bacteria. It is used for the coliform forming bacteria. So here production of acetyl methyl carbinol is used for the 𝙂𝙖-Pascal test. Option A is the correct answer. So positive reaction, a pink red color on the surface for the Listeria. Negative reaction, a lack of pink red color is for the Streptococcus mitis. Many bacteria ferment this carbohydrate with the production of acetyl methyl carbinol called acetoin. This question was asked in GPAT 2020. Okay, so this question is very, very important. Please note it down. Next question. To test the indole production. Okay, indole production. Yes, if this question is also related to this IMBIC test. Okay, to the test of indole production, the peptone water culture, which reagent is used? Salon-Waskis reagent, iodine potassium iodide reagent, Benedict's reagent, या Kovacs reagent? Answer this question. I want answer from each and every student. Okay, I'm checking the chat section. The chat section is quite silent. I think no one giving the answer. The indole production test or the to check the whether that particular bacteria is producing indole or not, we need use a specific reagent called Kovacs reagent. Okay, Kovacs reagent. Option D is the correct answer. Okay. So Kovacs reagent, this is used for the detection of indole producing bacteria by using a enriched reagent or Kovacs reagent. The formation of red or pink colored ring at the top is taken as positive. This test gives a positive for E. coli and negative for Klebsiella. Next question. Which of the following is an enriched medium? So enriched medium is a classification of functional type of culture medium. Functional type of culture medium. If we will talk about this, they are either blood agar, Lostin Janssen, Robertson Cook medium, or selenite F block. So you have to identify which EJ comes under enrichment medium. Which of the following is a enrichment medium? So the correct answer here is selenite F broth medium. So if you see, we have classified the culture medium on the basis of many types, functional type, whether it is based upon the physical state, solid, solid or liquid. So like that we classified. So here, so depending upon the functional type, we have classified as enriched, enrichment media, selective media, differential media, incubator media, sugar media, transport media, assay media, and storage media. So enriched media example is selenide F broth media. Selenide F broth media. Okay. So here option D is the, sir, when the next RRB notification is expected. See, so the new notification for the RRB has not been updated till now. So we are expecting the in the calendar it might be updated in next 2 to 3 months. So you are expected to see in 2 to 3 months. So my advice is whether the exam will of the notification has out or not. The thing we should be focusing on our preparation, how our preparation is going on, whether our preparation is up to the mark or not. So whether we 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 always keep our preparation should at up to the mark. Whenever the notification will become, you will ready to attend. So that is the difference created by GDCN. So GDCNs never wait to the notification will out and we will start preparing. No. So what our judicial is doing, we always keep our preparation to the up to the mark. Whenever the exam, whether it is a GPAT, whether it is sniper, whether it is pharmacist, drug inspector, whenever any exam will come, we will ready to fight. Okay. Chalo. Moving to the next question. The principal structural component of the cell wall in the bacteria is made from simple protein, peptidoglycan polymer, complex polysaccharide, or glycoprotein. The principal component of the cell wall is basically peptidoglycan. Okay, the cell wall is basically peptidoglycan. Option B is the correct answer. Next, you can see bacteria is a unicellular prokaryote. The cell wall is basically made from peptidoglycan. Okay, next question. The acidic polymer of ribitol or glycerol. The acidic polymer of ribitol or glycerol phosphate present in gram positive microorganism. So first thing is, when the question is about gram positive microorganism, so gram positive microorganism always contains echoic acid or lipoechoic acid. If the same question is about gram negative, then your answer will be lipopolysaccharides. Okay. So here the answer is option B. Okay. Next question. For the gram negative, it contains lipopolysaccharide. Now you have to match the list 1 with list 2. Okay. You have to match the list 1 with list 2. So here you have classification of the bacteria. Here you have exam. Gram positive spherical shaped non-motile bacteria, gram positive sporulatic. Okay, let's discuss. So gram positive spherical shaped bacteria. Example is your Streptococcus species. Gram positive sporulating obligatic anaerobic bacteria. Obligatory anaerobic bacteria is your Clostridium tetani. Okay. A gram positive rod shaped non-sporulating bacteria is your Corynebacterium diphtheria. Okay. Next is gram positive sporulating rod shaped motile bacteria is your Bacillus anthracis. So here option C is the correct answer. Okay. Option C is the correct answer. Streptococcus species, it is a gram positive facultative obligate aerobe. Clostridium tetani, it is a gram-positive bacillus obligate anaerobe. Corynebacterium diphtheria, it is a non-capsulated, non-sporulating gram-positive rod. Bacillus anthracis, it is a rod-shaped bacteria and motile bacillus. Okay. Next question. A microscopic examination of culture isolated revealed that there is a spherical bodies with a smooth outline growing in the long chain. Identify the microorganism. What it is telling that a microscopic examination of culture isolated revealed that spherical bodies with a smooth outline growing in a long chain. Identify the microorganism. Whether it is a Streptococcus aureus, Streptococcus pyogenes, Rhizococcus stolonifer, या Bacillus subtilis. Here, the correct answer is Streptococcus pyogenes. Okay, so Streptococcus pyogenes, you can see this is a Streptococci family divided in one place, forms a chain-like structure. Okay, you can see the chain-like structure will be seen. Next question. Size, shape, and mode of arrangement in is a typical of certain microorganism. Match them correctly. Streptococci, Saricna, Bacillus anthracis, Vibrio, Spirilla. Okay? Huh. You have to match. So what you have to match? So Streptococci is a gram-negative positive arranged chain. Saricna is a multiple of 8. Saricna, you know, 8 cells like this. 1, 2, 3, 4. Again, at the back side, 1, 2, 3, 4. Bacillus anthracis is basically a rod-shaped acid-fast bacteria. Vibrio and Spirilla. Vibrio and Spirilla are the comma and A set. That means your option A is the correct answer. Okay? Your option A is the correct answer. Next question. Next question. So you can order for to increase your preparation. So you can also get this book that is GPAT at your fingertip book, which is basically the summarized questions of divided of each chapter question with their detailed explanation along with the theoretical book. So this is the one of the most trusted book by the toppers. So it is consisting of all the modules in a summarized, well uh, documented in a colored format. It will easy for reading. Okay. Theory is 899, objective is 749. Next. So you can, whatever the classes, live classes is going on in your YouTube, you can attend the same with the in our app. So to joining that app, you are joining that batch, you need to open the GDC classes, go to the batch section, you have to apply the code MAHA26. So MAHA should be in small letter and 26. So after that, you will be added to that batch. Once you have added, you can give the exam over there. Okay. Next question. The optimum temperature for rapid growth of mesophiles. So based upon the temperature, bacteria are classified to three classes. Okay. Psychophil, mesophil, thermophil. If we talk about mesophil, it should be 25 to 40 degrees. If the question is about psychophil, it is 0 to 15 degrees. And if it is thermophil, that is 45 to 65 or 50 to 60 degrees. So here option C is the correct answer. 45 to 70 degree is for thermophile, mesophile it is 20 to 40, psychophile 0 to 50. Next question. For thermophile is, I told you for psychophile it is 0 to 15 degree, for mesophile it is 25 to 40 degree, for thermophile it is 45 to 70 degree. So here the answer is 45. So option C is the correct answer. Okay. Here the correct answer is option C. Next question. Obligate anaerobes. So obligate anaerobes means these are the bacteria which dedicatedly grow in the absence of oxygen. Okay, dedicatedly grow in the absence of oxygen. So let's find the correct answer. Can tolerate oxygen and grow better in presence? No. Do not tolerate oxygen, die in its presence. Can grow in oxygen level below normal. Can grow in the presence of atmospheric oxygen. So the correct answer is option B. If it do not tolerate oxygen, it can die in its presence. So option B is the correct answer. Obligate anaerobes are grow only in the absence of oxygen, and obligate aerobes are only grow in the presence of oxygen. Okay, next question. The category of bacteria comprising of methogenic bacteria, extreme haploids, and thermoacidophilic are grouped under which of the following? Methogenic bacteria, extreme halophiles, and thermoacidophilic. So they are grouped under which class? They are grouped under Archaebacteria. Okay, they are grouped under Archaebacteria. Next question. Example of Archaebacteria. Methogenic bacteria extremely environment grow in extreme environment conditions. These organisms are grouped under either Archaebacteria. Example of Archaebacteria is listed over here. Next question. Which of the following is classified based upon the staining technique? Okay. And this question was asked in GATE 1993. Okay. So let's match. So you can see Clostridium tetani. So Clostridium tetani is a gram-positive bacillus. Okay. E. coli, you know, gram-negative. It is a gram-negative bacillus. Neisseria gonorrhea. Uh, gram-negative. E. coli is a gram-negative bacillus. Neisseria gonorrhea is a gram-negative cocci. Streptococcus pyogenes is a gram-positive cocci. That means 1Q, 2S, 3R, 4P. Option D is the correct answer. Okay. Next question. So GPAT ranker batch has been started. So it has recommended to the best course to revise all the important content in a short period of time. So this class is featuring around 200 plus live classes, 500 plus online test series, rapid revision ebook, 25 class tests, and more than 1,000 recorded video. So this batch has been started, or it is highly recommended batch by the toppers. You can ask your seniors, those who have qualified GPAT, with the help of GTC and other. You can ask them; they will definitely recommend this batch. If you want to revise the course in a short period of time, this is highly recommended. Chalo, moving to the next question. Given two below are the statement. One is assertion, one is region. Exotoxin diffuses freely through the bacterial cell wall into the medium which each organism is going. Yes, it is correct. They are water soluble and can pass through the surrounding medium. Yes. So actually what happens, exotoxins are water soluble. Due to this water solubility, they can diffuses freely through the bacterial cell wall into the organism medium. So option A is the correct answer over here. Next question. The most active stage in the sigmoidal curve of bacteria which has maximum growth. You will see time and growth. Thus, so first page is, it is like this. This is your lag and this is your log phase. So log phase, your maximum growth is it. Here you get maximum growth. Okay. What you get here? You get maximum growth. Next question. Which match the process of reproduction and genetic exchange under column 1 with the explanation under column. Binary fission. So you know binary fission is the most common vegetative reproduction process. Transformation is the gene transfer by directly taking of the foreign genetic material to the naked DNA. Transformation through a virus, that is bacteriophage. Conjugation by the formation of a cell contact or called cytoplasmic bridge. So here 2 1q 2s, option D is the correct answer. So transfer of the, this is a transferring method introducing foreign DNA into the bacterial cell. Conjugation, during conjugation process, two live bacteria, donor and recipient, come together joined by a cytoplasmic bridge. Transduction, that is introduction of DNA into the host cells. Okay. And transcription, you know, transcription is the process conversion of formation of RNA from DNA is called as transcription. Binary fission is the most common method of bacterial reproduction. Okay. Next question. Which of the following microbial form listed below exhibit highest level of resistance to physical and chemical method of control? Okay, which of the following will give highest level of resistance? So highest level of resistance is basically produced by your naked virus, protozoan yeast, vegetative bacterial cell, and endospore. So this is very, very important. This question was asked in GPAT 2021. Okay, it is actually given by this bacterial endospore. Bacterial endospore will provide highest level of resistance. During unfavorable condition, bacteria will produce this endospore, and this endospore will be live like a dormant. Whenever a favorable condition will be arise, it will reproduce and regenerate to a whole bacteria. Next question. Which of the following is the causative organism for syphilis? So syphilis, Clostridium tetani is causes tetanus. Bacillus pertussis causes whooping cough. Treponema pallidum causes syphilis. Vibrio cholera causes cholera. So here the correct answer is option C. Okay, very good. Option C is the correct answer. You can follow this table. This is there in our Navigator series module number 14 at page number 149. Okay. Next question. The most common causative agent for bacterial pneumonia. Okay. Most common causative agent for the bacterial pneumonia is Streptococcus pneumoniae. The causative organism is Streptococcus pneumoniae. Option C is the correct answer. Okay. Streptococcus pneumoniae is the correct answer. Next question. Match the following under column 1 with the respective causative organism. This is also very simple, direct question. This question was asked in GPAT 2018. So first thing, syphilis, everyone know? Syphilis means Treponema pallidum. Plague is Yersinia pestis. Typhus is by Rickettsia prowazekii. Creutzfeldt-Jakob disease by prions. Okay, that means 1Q, 2R, TS. Option C is the correct answer. Okay, very good. Next question. You can follow this table. This table is extremely important. You can write it down. Okay, you can also, this table from this question can be asked. Next question. Roll tube technique is the modification of pour plate technique, straight plate technique, micro manipulator technique, and spread plate technique. So roll tube technique, this is the method of spreading microorganism on a culture medium. So roll plate technique is basically the modification of straight plate technique. Okay, option B is the correct answer. So this is the roll tube technique is the modification of straight plate technique. This question was asked in GPAT 2019. Same thing is there in our module, page number 151. You can refer to this. Okay. Next question. Which of the following is a acellular? Very, very important question. Recently, this question was asked. So, acellular organism is a virus. Okay. Virus is a acellular. It does not have any cells. That and virus is called that this is a connection between living and non-living organisms. So virus is a connection between living and non-living organisms. Bacteria, fungus, virus, and ammonia. Your correct answer is virus. Virus does not have any cells. Next question. Which of the following is a obligatory intracellular parasite? Which of the following is a obligatory intracellular parasite? Or virus, fungus, mycobacterium, rickettsia. You have to find. So please remember virus and rickettsia. So these are the species, they only depend on a host to grow. So outside they do not have any life. Whenever it enter into the host, it use the host body mechanism system as its own system for their growth and development. So virus and rickets are the considered obligatory and intracellular species. Okay, these are the parasites. They cannot reproduce outside their host cell. Okay, they cannot reproduce. When it enter into the host cell, it gain the ability it can reproduce. Obligate intracellular parasite in human include viruses, certain bacteria like Alamidella, Rickettsia, Coxilla, and certain species like Mycobacterium leptre. Next question. So GPAT Mahamarathon book third edition has been released, which became the one of the bestseller book. You can purchase. The price is only 599 rupees. Okay, this is has all the previous year question with detailed explanation. Okay, you can purchase with the colors check. You can purchase. Viroids differ from virus. Viroid differs from virus have in having naked DNA molecule, satellite RNA packaged with viral genome, naked RNA molecules, and naked DNA packed with a viral genome. Yes. What is viroids? Viroids are nothing but they are having naked DNA molecules. Okay? Sorry, naked RNA molecules. Sorry. Viroids have nothing but they are naked RNA molecule. And these RNA molecule is act as the nucleic acid. When it enter into the host, they inject their RNA inside the bacteria where there is a reverse transcription will be there. That bacteria now from the RNA, the viral DNA is formed. That viral DNA has the ability to incorporate it into the host genome and produces viral protein. So this is a complex mechanism which has exerted by a virus. Okay, this is a complex mechanism exerted by a virus. So viruses are infectious, non-protein coding, highly structural, small circular naked RNA to replicate autosomally induces disease in higher plants. Next question. Which of the following statement is incorrect about characteristics of virus? Incorrect. Okay. Virus is lack of enzyme for protein and nucleic acid synthesis. Most animal viruses are spherical in shape. Virus is not cellular, but there is a presence of nucleus. bacteriophage, virus that parasite on bacteria. You have to find the incorrect. So virus is lack of enzyme. Yes, correct. Most animal virus have spherical shape. Yes, it is correct. Bacteriophage, that virus. Bacteriophage is the virus that has ability to infect the bacteria. But please remember, virus is a acellular, does not have cytoplasm and nucleus. So please note it down. Option C is the correct answer here. This is the lack of enzyme, protein, and nucleic acid. They have lack of nucleus, cytoplasm, and cell membrane. Okay. Most of the animal viruses have spherical structure. Bacteriophage are the virus that grow on the parasite, like parasite on a bacteria. That means this is a virus which has the ability to infect a bacteria. Next question. Influenza virus. Influenza virus are the RNA virus and belonging to which family? Orthomyxiviridae, Papoviridae, Retroviridae, or Parvoviridae? This question was asked in GPAT 2014. Influenza virus is a which family? It is belongs to which family? Orthomyxiviridae family. Okay. So it already this table has there in the module. You can refer page number 115. So double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded positive RNA, single-stranded negative RNA, RNA reverse transcriptase, DNA reverse transcriptase. These are the group of virus and their family. Next question. Infected blood products may produce serum hepatitis. Infected blood products may produce serum hepatitis due to the presence of hepatitis A virus, hepatitis B virus, hepatitis C virus, or none of these. Answer this question. So please remember, so the infected blood and other serum product is basically occurs due to this hepatitis B virus. Okay, this is transmitted through this one. Next question. You can see serum hepatitis transmitted through infected blood products. Very, very important point. Next question. Incorrect statement about the important properties of coronavirus. Non-enveloped RNA virus, enveloped RNA virus, contagious, can spread from person to person. Okay. So here you have to find the incorrect statement. Okay. Whether it is, so if you have seen the structure of the coronavirus, it looks like this. Spikes all over the spikes in the body. And after this spike, there is a layer like this. So that means they are enveloped. There is a outer covering is there around the virus. So here non-enveloped RNA virus is incorrect. Rest of the three statements are correct. Here the question is about incorrect. Okay. So this contains single-stranded positive sense-strand RNA virus. Okay. This is the enveloped RNA virus. It is highly contagious, can through respiratory droplet, aerosols, and close contact spread from one person to another person. Next question. Neutralization test is generally used for the serodiagnosis of virus, bacterial infection, fungal infection, and protozoal infection. The neutralization test is basically done for the virus. Okay. So neutralization test is the antibody neutralizes viral infectivity. Plaque assay, this is a gold standard technique. These are the all are virus detection technique, which consisting of plaque assay or which is the gold standard technique, hemagglutination test, hemagglutination inhibition test, and serological methods. Next question. Which of the following about the BZB virus is not true? Which of the following about VZV virus is not true. Varicella zoster virus developed after individual exposed to varicella zoster virus for the first time. Here you have to find which is not true. Okay. Herpes zoster develops from reaction of the virus later life reactivation. There are no vaccine for this virus. The infection result in post-hepatic neuralgia. We have to find the not which is not true. So VZ for VZV virus, we have vaccination. Okay, your option is there no vaccine. That's why this option is incorrect. Next question. Varicella zoster vaccine, virus vaccine, also known as chickenpox vaccine, protect against chickenpox and shingles. Next question. Which of the following is not true about Ebola virus disease? This question was asked in 2018. Not true. Okay. Spread through human to human contact, human transmission by direct contact. Antiviral drugs are approved by FDA to mitigate the infection. Diagnostic test includes ELISA, the virus named after the river, which is incorrect. For EVD, Ebola virus disease, there is no antiviral drug has been approved by FDA. Okay, so here question is antiviral drug are approved by FDA to mitigate infection. No, no antiviral drug has been approved. We are giving as a supportive treatment. Okay, we are giving basically supportive treatment. No specific treatment has been there. No antiviral drug are approved by the FDA for the mitigating this infection. The etiological agent of the infectious mononucleosis are associated with a form of Burkitt's lymphoma is VZV, Epstein-Barr virus, picornavirus, या papovirus. Burkitt's lymphoma. The virus that can induces cancer is this Epstein-Barr virus. So Epstein-Barr virus can induces Burkitt's lymphoma. Okay, what it can induce? It can induce Burkitt's lymphoma. Infectious mononucleosis is caused by Epstein-Barr virus. It is a double-stranded DNA virus of the herpes virus family. So this EBV infection infect basically the B lymphocyte CD21 receptor. Okay, last question. This fungus is also known as opportunistic fungus, opportunistic pathogen, often isolated from a warm-blooded animals. Okay, opportunistic fungus means these are the fungus. They are grow if the host immune system is compromised, it has easily infect that host cell. The opportunistic pathogen or opportunistic fungus here is your Candida albicans. So Candida albicans is called as opportunistic pathogens or fungus. Candida is aid to digest the nutrient absorption. This fungus is known as opportunistic pathogen isolated from warm-blooded animals. Okay. So whatever we have discussed in the today's class, you need to go and join the batch. The code for this batch is MAHA-26. Okay. The batch is MAHA-26. So all are in small letter. You have to join that batch. Once you join, you need to attend the test, because unless until you will not solve many questions, you will not get enough confidence for your GPAT exam. So that is the reason when you practice many questions, you get more confidence for your upcoming exam. Okay, this is my advice to join the batch and give the test regularly for your until your GPAT exam over. Okay. How many questions can be expected from microbiology? See, from biochemistry, biotechnology, and microbiology. From these three questions, every year around 10 questions they ask. Okay, the number varies like 30%. Okay, 10 to 12, 10 to 11 question you will see from these 3-4 questions from biochemistry, 3-4 questions from microbiology, 2-3 questions from biotech. Okay, so I think all the questions that we discussed today is clear to all of you. If you have any questions, so you can ask. Okay, so that's all for today. Thank you. Thank you so much for joining this class to boost your revision with GDC. So stay connected with GDC for upcoming Mahamarathon lectures. Okay. Thank you. Thank you so much.